Very good morning students. Sai Ram. Welcome back to our science period. Children, you know very well, yes, now I am going to continue this chapter, that is Introduction to Biotechnology. Okay, so can you remember children, last session what we discussed? We discussed about permanent tissue. How it is produced, the permanent tissue, how it is produced, and yes, what are the types of permanent tissue? There are two types of permanent tissue. First one is what? Simple permanent tissue, and the next one is complex permanent tissue. Okay, simple permanent tissue, there are three types, that is parenchyma, collenchyma and sclerenchyma. Complex permanent tissues, xylem and phloem. Okay, xylem and phloem. So children, these permanent tissues, where they are located and what are the function of these tissues, these all things already we discussed, isn't it? So I hope you read your textbook also. Yes, very good. So children, today I will start with one new part, that is biotechnology. Okay, this is one of the, yes, very important chapter. Okay children, biotechnology. See here, biotechnology helps to produce plants and animals with some new characteristics along with their natural ones. Okay, so biotechnology means what, that I will tell you. Bio, just you have to say here, bio means what? Living things. Okay, technology. So that one technology that we are using, yes, the technology is used with the help of that living organisms, that is called what? Biotechnology. So what is the importance of this technology? It helps to produce plants and animals with new characteristics, okay, the desirable characteristics, so that is along with their natural ones. For example, capsicum. So nowadays we are getting that colorful capsicum, isn't it children? Instead of red is the natural one. Yes, green and yellow also we are getting. So that is with the help of this biotechnology. Okay, that is the hybridization. So with the help of this biotechnology process only, we are getting different colors of vegetables. For example, that purple color cabbage, that is also one of the result of this biotechnology. Okay children, so see here, that is capsicum, already I told, that is new variety, that is called what? That is red and yellow color. So it is very attractive, at the same time nutrition value of that capsicum also high. Okay, so see here children, the biotechnology means this is one technology that is used in the technology that living organisms are used. Okay, that's why it is called what? Biotechnology. What is the necessary, what is the importance of this technology? Yes, with the help of this biotechnology we can get that new characteristics of plants and animals along with their original one. Okay children, see here, the next one is the techniques of bringing about enhancement in living organisms using artificial genetic changes. So that artificial genetic changes we are giving with the help of the biotechnology process, and through hybridization for the welfare of human beings are together known as what? Biotechnology. Okay, so what is the importance of this biotechnology? Today we will discuss. Okay children, see here, there are two important techniques, that is two main techniques used in biotechnology, that is genetic engineering and tissue culture. Okay, so see here two important techniques, that is genetic engineering and tissue culture. So what is meant by genetic engineering and what is the function, you will study in your higher standard. Now introduction of tissue culture, that is in your textbook, that I will tell you. Okay children, so before that, you know very well what is meant by tissue. It's a group of cells. Okay, see here children, tissue culture. The tissues is the group of cells that already you know, having the same origin, same structure and same function. Plants and animals have different type of tissue for performing the necessary functions, that also we know, isn't it? So yes, have you seen that flowers of the same variety but of two or three different colors? For example, that hibiscus. So See here that 2-3 different colors born by the same plant. Have you seen? Yes or no? So see here. This is done using the modern gardening technique. That is what? Tissue culture. Okay. So see here the ex vivo growth of cells or tissues in an aseptic and nutrient rich medium is termed tissue culture. So the group of tissue is separated from the plant and it is grow. Yes, that is grow in a nutrient rich medium. Okay, that is called what? Tissue culture. Okay, so it's possible to develop a complete organism from a single cell or from tissue. So that complete organism we will get from single tissue or what? Single cell or the group of tissue by applying the tissue culture technique. Okay, so tissue culture technique means that tissue it is separated from the parent and it is cultivated in a laboratory with some specific medium. From the tissue we will get a new plant or animal. Okay, so children see here, this is a tissue culture technique. This is a source plant and here that is the banana, isn't it? So this is the bulb come from that source plant and here the tissue is separated and the tissue is treated the primary in the laboratory. Afterwards what? The reproduction or multiplication. Okay. And next one is yes, shooting and rooting formation. Next the primary hardening and next one is what? Secondary hardening. So this is what the tissue culture steps. So during this technique, a liquid, solid, gel-like medium made from agar, okay, is used. So that medium name is called what? Agar or broth medium. That is prepared from that some algae. Okay, children. So this gel-like substances supplies nutrients and energy required for tissue culture. So children, for this tissue culture, yes, the tissue wants the nutrients, isn't it? That separated tissue wants what? Nutrients. That nutrients they will get from one gel-like medium. That is called what? Agar medium or broth medium. So that medium supplies nutrients and energy to the tissue. Next, we will discuss about the changes in agricultural management due to biotechnology. Okay. First point is that is genetically modified crops, that is GM crops. So here GM crops are being produced by introducing changes in DNA of our natural crops. Normally such varieties are produced artificially. So genetically modified crops means what? It is produced by. So children, genetically modified crops that is produced by introducing changes in DNA. Okay, you know very well that plant cell and animal cell is made up of a DNA. The remaining cell inside the cell, that DNA, gene is there, chromosome is there, isn't it? The chromosome is made up of a DNA. So normally such varieties are produced artificially. Different useful characters are introduced in such varieties. Okay. Next one is what is the uses of that GM crops? See here, ability to withstand environmental stress. Environmental stress. Some naturally occurring varieties cannot withstand environmental stresses like yes, changing temperature, wet and dry, famines, changing climate, etc. So these are what? Environmental stress. So the natural crop they cannot withstand with the yes, these environmental stresses, but these GM crops can have a ability to withstand. Okay. However, GM, the genetically modified crops, can grow in any of such adverse conditions. Okay. Next one is as GM, the genetically modified crops are resistant to insect, pest, pathogens, chemical weedicides, etc. The use of harmful chemicals like pesticides can be avoided. Okay, due to use of seeds of genetically modified crops, there is improvement in nutritive value. Okay, children, and a decrease in loss of crops. So these are the, yes, beneficial of the genetically modified crops. See here, some of the genetically modified crops in your book. It is in your textbook. You have to read this one. Okay, children. Next one is, yes, why it is necessary this biotechnology. See here, in this way a green evolution is being achieved. Is achieved through tissue culture, and a country like India, that means India, the population is more, so we want more food, isn't it? So country like India with a huge population is meeting with success in producing sufficient food grades for all. Okay, children. So through this statement you will get that is the importance of tissue culture and biotechnology, isn't it? So we have that we want that India want that lot of food, because here the population is more, so that our food is fulfilled with the help of this tissue culture. Next one is, see here, application of biotechnology in floriculture, nurseries and forestry. In that we will discuss some points are there. So here first one is, nurseries are used to, yes, first that some information. Nurseries are used to grow gardens on large scale for afforestation, reforestation and so on. Seedlings are required on large numbers for all these purposes. Okay, which one is seedlings? So such cases, it is profitable to manufacture plantlets with the help of tissue culture techniques. So here first one is, the tissue culture is used to grow on large scale those plants that bear flowers, fruits of excellent quality. Okay, it is possible to get fully grown plants in shorter durations. This is second point. Next one is, so children, see here the next point, plants can be grown on a large scale even without pollination or if germinating seeds are not available. Okay, so you know that with the help of the pollination, that is one of the important process. Okay, so plants can be grown on a large scale even without pollination or if germinating seeds are not available. For example, that orchids or pitcher plants do not germinate but can easily be generated using tissue culture. This is another important thing. Next one is, large numbers of seedlings, plantlets can be produced in a short time using minimum resources and materials. Okay, children. Usually, plants produced by tissue culture and genetic modification techniques are disease free. Okay, this is another one of the important thing. Those plants produced by tissue culture and genetic modification, that is disease free. Plantlets manufactured by tissue culture of the meristem are virus free. Okay, next one is, embryos formed by conventional hybridization techniques between two or more varieties may not grow fully for some reasons. Okay, that which embryos formed by, yes, conventional hybridization techniques between two or more varieties, sometimes it may not grow fully for some reasons. But however, embryos produced by tissue culture techniques always complete their growth. Next one is, yes, rare and endangered species. Nowadays, we are having many that rare and endangered species. That means that is in a last stage. Some plants, that very few numbers of plants or animals are there. That is called what? Rare species. For example, that nepenthes plants. Okay. So that is that plants can be grown and protected from extinction by applying tissue culture techniques. Similarly, various parts and seeds of such plants can be preserved by tissue culture and those varieties can be protected. So nowadays that environmental stress is more. So for that some plants can't withstand. So they are in a rare species. Okay, that means they are in a last stage. Only few numbers of plants or trees or animals we are having such kind of species. We are saying that is a rare and endangered plants. Okay, so that is stored, preserved, that some parts of that plants or seeds are preserved with the help of the tissue culture. So when we want that time we can cultivate that plant. Okay children, so these are the important points of biotechnology and tissue culture. So today we discussed about what is meant by biotechnology and tissue culture. Okay, so children, you have to read your textbook. Now only little bit portion is remaining, that next class I will complete it. Okay children, thank you. Saira.